Hello everyone, I am Dr.K.Geetha, Assistant Professor, Department of Botany, the Standard Fire Oaks Rajaratnam College for Women, Sivahasi. So today we have to see the DNA as the genetic material. So under the scientist proved by as a Griffith experiment. Yes, so DNA is the necessary things for all living organisms. So today we have to learn as a Griffith experiment. Okay, so first you have to see the as a introduction part of Griffith experiments. So first I want to tell as a intro part of Griffith experiment. So in 1928, as a Griffith demonstrated that as a genetic information could be transformed, so without knowing so what this information was, so they can told as a transforming principle. So it was the first step towards the proving that the nucleic acid, that is nothing but that is a DNA, is essential for all the living organism. It has as a proteins and the chemical basis of experiments. So he was studying as a pathogenicity of as a different strains of bacteria, especially so he was used as a Diplococcus pneumoniae. So he studied the effect of two different strains of bacteria. So one is R2 type, second one as a S3 type. So in R2 type, so it has as a irregular rough colonies divide off as a mucilaginous sheath. So it is otherwise known as a non-virulent. So second type, that is a S3 type, so it has as a smooth colonies having mucilaginous sheath. So it is otherwise known as a virulent type. So in this experiment, as a Griffith used as a test animal, that is mice. So first type, that is a R2 type, he injected into mice. As a mice survived. S3 type, as a virulent gene, it may be injected into the mice. As a mice, it may be died. As a heat killed S type, heat killed, so nothing but as a virulent, it may be killed in particular pathogen. So it may be injected into a particular mice. As a mice, it may be surveyed. Surveyed means it can be live. So as a heat killed S type plus R2 type. So both genes are combined. So again it can be injected into the mice. So after that, as a mice, again it can be died. So these are the basic or fundamental phenomena for as a Griffith experiment. So this is the experiment, so we can done and we can do so this kind of work in already laboratory. So under the Griffith experiment, so did not produce that DNA was responsible for transformation. So he would design an experiment to prove that as a DNA has responsible for the transformation. So this is the conclusion, but not all the scientists are accepted this kind of report. So after that, Avery, McCarthy and MacLeod reported and again do as a Griffith experiment. So with the help of some pathogens, or they can use as a heat killed smooth type of bacteria. So this experiment is essential for to prove as a DNA is the energy transformation or gene transformation in all the living organism, especially for they can use as a mice. Thank you for visiting. So again we will meet you in next video. Thank you.